All right, happy Wednesday, 8th grade. You have everybody's favorite, reading and notes. So please make sure to complete the reading in the CPO Life Science textbook. You can find it online. The login for my class is Wriggle8. The password is WriggleSci8, Wriggle Science. You can find all that information on our Schoology page. All right, so these are the notes for Chapter 11, Heredity, Section 11.2, Predicting Heredity. If you follow along with my notes, copy them word for word, you are guaranteed a B on this assignment. If you include the summary at the end, it'll give you an A. So, super easy assignment today. Please make sure to keep up. First is our vocabulary terms. Punnett square. We talked about this a little bit in class the other day. For the previous section. A Punnett square shows all of the possible combinations of alleles for the parents. And so that was when we were crossing our V's on those flies, and it looks kind of like that. So I showed you that way with multiplying with FOIL, and then I showed some of you guys the box right here, which you can use instead. We're going to talk about those today. We're also going to talk about probability. Probability is the mathematical chance that an event will occur. How were genes finally discovered? So, obviously, way back in the day, they had no clue what a gene was. They barely knew, how you could tell who the parents are. So Maury did not exist. He didn't have his DNA testing. He could not have a show that says, the DNA test says that you are the father. So that didn't happen back in the day. So here's what happened. So in 1865, that Gregor Mendel guy that we talked about last week, he described heredity, but he did not know about a little thing called genes yet. It wasn't until 1902 when Walter Sutton observed chromosomes in grasshopper cells. He saw chromosomes in homologous pairs. They separated during meiosis. In 1903, Sutton found that chromosomes contained genes. It took almost 40 years to discover genes and finish explaining Mendel's description of heredity. So way back here, 1865, Mendel described heredity. It wasn't until almost 40 years later, in 1902, 1903, when there was a word that could be used to describe how heredity is passed on. Homologous pairs, if you remember, that's when your chromosomes are either just two like that, a pair of the same type of chromosomes, or they can be crossed in an X if we're talking about after prophase has happened and during mitosis or meiosis. How are traits passed on to offspring? So, individual units called genes determine an organism's traits. A gene is a segment of DNA located on chromosomes that carries heredity instructions from parent to offspring. For each gene, an organism typically receives one allele from each parent. So like I keep saying, half from mom, half from dad. You need two alleles to make a pair. Every chromosome comes in a pair when you're talking about the completed cell. If an organism inherits different alleles for a trait, one allele may be dominant over the other. The alleles of a gene separate from each other when sex cells are formed during meiosis. So what happens to alleles during meiosis? Alleles of a gene separate during meiosis. Alleles are found on chromosomes. Homologous pairs of chromosomes separate in meiosis too. How do the alleles separate? Well, let's start with a cell right here. It should have probably more than 2 chromosomes in it, but we're going to keep our life simple. We're only going to look at 2 chromosomes for now. We're not going to look at all 10, 20, 30, 40, 50, however many there should be. We're only looking at one to make our life simple. So I have one chromosome here. It's going to represent a dominant allele. And a green chromosome over here. It's going to represent a recessive allele. So on each chromosome, there's all sorts of little pieces of information. And each piece of information is going to be a little bit different. But they're going to be talking about the same topic, I guess we could say. So maybe this is telling me the flower color. And this one is right here. It says the flower color is going to be white. On this one, maybe it says that it's going to be blue. Or green. There we go. We'll have a green flower and a blue flower. I don't know of very many green flowers, but they're out there. So on this one chromosome, it might say the flower color. It might also say if the seeds are going to be round or wrinkled. It might say a whole bunch of things on this chromosome. But in that chromosome, there's a bunch of little pieces. And each piece is going to be an allele. So we're just going to look at maybe this allele right here, these two dark ones. So we have our cell. It's going to undergo meiosis. So these chromosomes are going to double. So now I have 2 blues and 2 greens. They're going to go through that first stage of meiosis 1, and they're going to split. The blue ones go over here, the green ones go over here, but they're still paired up. In meiosis 2, when they finally create their haploid cells in their cytokinesis stage, we're going to end up with one blue chromosome, and these I said were the dominants, and then this one can only give one of each of the recessives. So we have 4 sex cells created, and the alleles are now separated. Okay, so let's come back. In the original cell, it had one of each type of allele. It had a dominant and a recessive. After it undergoes the 2 divisions of meiosis, because you remember, 2 times cells divide, half the chromosomes, it's called haploid. Oh, I'm spitting fire. So after it splits up into its 4 sex cells right here, there's one of those chromosomes each. They're split up. Fertilization occurs when sex cells from 2 parents combine. The offspring will inherit one homologous chromosome from each parent. In other words, half of each homologous pair comes from each parent. So a homologous chromosome means 2 chromosomes of the same type. It doesn't necessarily with the same exact information on them. It doesn't have to have the same alleles, but it still has to be the same type of chromosome. So maybe it's the 2 chromosomes that would tell us your eye color. Maybe it's the 2 chromosomes that would tell us your hair. The 2 chromosomes that tell us how tall you're supposed to grow. Those would be homologous chromosomes, but they can have different information on each one. Okay, is there a way to predict the genotype and the phenotype? Yes. How can we predict genotype and phenotype? So we can use a Punnett square. Punnett squares can be used to predict genotype and phenotype if you know the genotype of the parents. So if we think back to 11.1, talking about Mendel and his pea plant experiments, in his first crossing of plants, true breeding purple and true breeding white pea plants were crossed. Up here, I have my purple plants. They're capital P, capital P. Over here I have my white flowers. They're going to be a lowercase p and a lowercase p. In order to fill out this Punnett square, I'm going to take my capital P here, and I'm just going to move it into both of the slots directly underneath it. I'm going to do the same thing here. Directly straight down, directly straight down. Now I'm going to take my lower cases. I'm going to move them straight across. So my P is going to go here, and my lowercase is going to go there. My lowercase there, and there. Not too bad, right? So you're just basically, you make a big empty box, and then you drop your letters in, or you pull your letters over. And so in each of these boxes, I have a capital P and a lowercase p, because that's the only information I could have. So it's just like our schedule. It's just like any table. You just take the information and you see where they meet up, except we're filling in the information based on what we have. Not too bad. So here's my list of all the offspring. Capital P, lower p, capital P, lower p, capital lower, capital lower. So all of the offspring have one dominant and one recessive allele. They are all heterozygous for purple flowers. So they all have a pair of different alleles, capital P and lowercase p, purple and white. So they're heterozygous, but they're going to have purple flowers because purple is dominant. So all of the offspring are capital P, lowercase p. So they are all purple. With a Punnett square, you can predict the probability for each possible genotype and phenotype of the offspring. So Punnett squares and probability. In Mendel's experiment, he allowed first generation capital P, lowercase p flowers, or heterozygous for purple, flowers to self-pollinate, and some white flowers appeared in a 3-to-1 ratio for the second generation. So if I take my capital P, lowercase p, from before, then I'm going to get a capital P, lowercase p, lowercase p, capital P, lowercase p, lowercase p. So after I fill it all in, I have capital lower, and capital lower, so 2 of those. And then I have one set of just my lowercase. These 3 all have a capital P in them. They are purple. These 3 all have the dominant trait. They will show the dominant trait. They will show purple. This one right here only has the recessive trait. It's only the lowercase. That means it's going to display the recessive trait, which is the white flowers. Okay, so probability. Probability is the mathematical chance that an event will occur. Probability can be expressed as a fraction or percentage. When you flip a coin, there's a 50% chance to get heads and a 50% chance to get tails. How a coin lands is completely random, unless it's fixed, cheaters. Just like a coin flip, inheriting genotype is random. Punnett squares represent all possible genotypes of offspring. In the previous Punnett square, 1 out of 4 squares is lowercase p, or 1 out of 4 can be written as 1 out of 4, 1/4. To convert this to a percent, divide the numerator by the denominator. Numerator is up top, denominator is down below, D for down, then multiply by 100%. So 1/4 times 100% is what? Well, first we're going to do that 1/4. 1 divided by 4. Let's get some decimal places here. I already know I'm going to need 2 decimal places. How many times does 4 go into 1? It doesn't. So I'm going to put my 0. I'm going to bring up my decimal place. How many times does 4 go into 10? It goes twice. So I'm going to put a 2. 2 times 4 is 8. So I'm going to subtract 8. 10 minus 8 is 2. 2 is less than 4. We're on a good track. I'm going to bring down this 0. How many times does 4 go into 20? It goes 5 times. 5 times 4 is 20, so I'm going to subtract 20 from 20. I get 0. So 1 divided by 4 is 0.25, or 25 hundredths. So I'm going to write here 0.25 times 100%. I have 2 zeros. I'm multiplying by a power of 10. All I have to do is move that decimal place over to the right 2 times, and my answer is 25. I'm going to pretend this is a unit, percent. So 25% is going to be my answer. So there is a 25% chance of offspring having lowercase p genotype. Well, what is the probability of getting purple flowers instead? There were 3 out of 4 squares that have a purple phenotype. 3 out of 4 squares have a purple phenotype. Their genotypes are either double capital P or one of each heterozygous, capital P, lowercase p. And there were 3. Let me come back here. So capital P, capital lower, capital lower, capital, capital lower, capital lower. So 1, 2, 3 out of 1, 2, 3, 4. 3 out of 4 can be purple. 3 out of 4 squares has purple. So I have 3 out of 4 times 100%. If I do my division again, it would be 3 divided by 4. And I'm going to need to do the same exact stuff. 7 times 4 is 28. I'll do this one a little faster because I'm not the math teacher today. So 0.75. 3/4 times 100% is the same as 0.75 times 100%. Again, I'm multiplying by a power of 10. There are 2 zeros. I can move my decimal place twice. 1, 2, I would have 75 point. So 75% is my answer. All right, the last thing you need to do, you need to write a summary. In order for you to receive full credit, you need to write a summary at the end of your complete Cornell notes. If you have copied my notes, then you already have questions over here on the left, notes right here in the center. You've already got it most of the way done. You're just missing your summary right now. So write your summary. It should be a few sentences about the main idea and the most important information. Get that done. Get your easy 100% on this homework assignment, and think critically about this work. Be ready to ask some questions. Tell me the stuff that you need more help on. I can't read your minds, and you guys can't read my mind either. So I don't know what you're missing unless you tell me. So please, please, please read these notes. Pay close attention. If you're having trouble with these ratios, please let me know. If this Punnett square stuff makes no sense, let me know. We are going to do more of it when we get back into Study Island, so you will get some practice with this. If meiosis is still a thing for you, that's still difficult, go back to Chapter 10. You need to finish your homework probably. And I will see you guys in class on Thursday.